So to start analyzing these data, these trait values that we've sampled from our hypothetical population, the first model we're going to assume is a single normal model. So in other words, we are going to assume that the trait values in our population are well represented or well captured by a normal distribution, a single normal distribution that has one mean and one standard deviation. Now this is a reasonable starting point for many traits in many populations because we know that continuous traits are often polygenic. They're controlled by many different genes, and because of that, they tend to have normal distributions. Now, as I mentioned above, we're going to assume that we haven't done the visual inspection and we don't yet know that these data are bimodal. So again, also for the sake of brevity, we're not going to talk here about the actual fitting of this model and how we conduct MCMC. But if you're interested, again, the trait values can be found in data.txt, and the MCMC for the single normal model can be found in the mcmc_singlenormal.rev file. And then the results of this analysis can be found in the single normal posterior.log file. So figure 2 here shows some of the parameter values that were sampled for the mean and standard deviation during the MCMC analysis that we've already conducted. So on the x-axis here of this plot, we have different possible parameter values for the mean of our normal distribution. And on the y-axis here, we have the standard deviation, possible values of the standard deviation. And so as you can see, we have our posterior surface here, which is represented by these lines. So each of these concentric circles here represents one sort of elevation in our posterior landscape. And so you can see that our MCMC analysis has sampled a series of parameter values, but they all tend to fall in roughly the same part of parameter space. So we have a mean that's maybe at or slightly below 4 and a standard deviation that's close to 1. So we can also look directly at this log file. Whoops. So there's our data. We'll get to that file later, sorry. So here's our log file. And so you can see, as with most log files from most MCMC analyses in RevBayes, we have on the far left, we have our iteration. So we've sampled every 10 iterations or 10 generations. You can see our posterior density, our likelihood, and our prior. And then in the fifth column, we have the sampled values for the mean. And in the sixth column, we have the sampled values for the standard deviation. All right, so going back to our tutorial. Whoops, sorry. So these, again, these red dots represent the combinations of parameter values we've sampled during MCMC. And if we just took one of those red dots and we actually plotted the normal distribution implied by those parameter values, it would look something like this. They all look fairly similar. So here we see that we have the single normal. This is the model we've assumed. And our MCMC analysis has suggested that the mean falls at or slightly below 4, and that the standard deviation, which determines the width of this distribution, is about 1. Now, just looking visually, it should be obvious that there's some very significant and important differences between the model we've assumed, represented by this black line here, again, the single normal distribution, and the bimodal trait data that we've actually collected from our population. But again, in many cases, doing this kind of visual comparison isn't going to be nearly so easy. And so we'd like to have a way to actually quantitatively compare the fit of this model to these data.